All right, good morning. Welcome, everyone. So before we get going, are there any questions over anything we've covered so far or last week or anything like that? Right, good. I think I've caught up in chat. I think I've caught up with most of the grading. I did grade a couple of the chapter summaries, and it looks like there may be one or two more that were turned in more recently that I need to get to. So I'll be working on those. Just as a reminder, the COVID training module needs to be completed. Otherwise, I will not be able to let you on campus. And there are several people who have not completed the test your knowledge quizzes for the COVID training. So that is, again, that is just a mandatory thing that everybody needs to do. You're only going to need to do it once this semester, but please get that done. Looks like there are 3 people I see that haven't completed that yet. So I'll just put out a reminder there, since we don't have to worry about getting on campus right now. So this is a great opportunity to do that. All right, good. So I think what I'll do is I'm going to spend a few minutes reviewing the quiz from last week, and then I'll pivot into talking about what we want to get into today. So I'm just going to go ahead and stop recording. Now that we've gone over the quiz, we're going to go and pivot into talking about lecture proper. So there are a couple of other interesting details that I want to get through, and then we will pivot into talking about reactive oxygen species, some metabolic pathways, and what happens when these pathways go wrong. And I provided some material for you all, a little video overview of most of the major relevant metabolic pathways, and then there was a little reading assignment regarding reactive oxygen species and free radicals. And so we will be integrating all of that and putting that together and making some more sense of it here today. So before we do that, however, I do have a question I want to ask you all. And the question is the following: are you all familiar with dextrose? Are you all familiar with dextrose?
Yes.
What is dextrose?
Sugar.
It's sugar. So let me ask you a question. Let me ask you all a question. Why don't we just call it sugar?
Because it's manufactured to match the body synthesized glucose.
Yeah, so you're moving in the right direction. So we say dextrose. Dextrose belongs to a class known as the sugars or the carbohydrates. And there are many different types of sugars. And so maybe dextrose is a type of sugar that the body can use, and that is true. Dextrose is actually something called glucose. And there are many other types of sugars like fructose and galactose and maltose and fructose. And some of those we can use and some of those we cannot use. But here's the question. Why don't we just call, when let's say we're going to give some dextrose 50 or some D50W to somebody who is hypoglycemic, why don't we just call it glucose? Because it is glucose. Why go through the trouble of calling it dextrose? I don't understand that. What's the deal? Does anyone happen to know what the deal is there?
Isn't the chemical makeup a little bit different from your regular sugars?
What do you mean? What do you mean by that?
Well, like you said earlier, they're many different sugars. It's just a different form of sugar that the body can process easier.
You're moving in the right direction. So I guess the question is there a difference between glucose and dextrose? What do you all
Isn't
think?
a little bit more complex sugar and dextrose is a simple sugar?
No, not quite. I think there's some intuition there that there's something going on. So here's what's going on, and this is important from a pharmacology standpoint as well. So dextrose is in fact glucose, right? And in fact, the chemical formula is exactly the same. And does anyone know what the chemical formula for glucose is?
C6D.
Yep. C6.
H.
It's a 2-to-1 ratio. So
it's 12.
there you go. C6H12O6.
Yeah, 06.
Yep, C6H12O6. Yep. So that's the chemical formula for dextrose. And dextrose, we can visualize it a couple different ways. It has 6 carbons. So you can kind of visualize the carbon structure kind of like this. But what I'm going to do is I'm going to unzip this ring structure and I'm going to draw it as a line. And the reason that we call dextrose is very subtle, and it does not have anything to do with the chemical formula. The chemical formula of glucose and dextrose is exactly the same, C6H12O6. What it has to do with is a concept known as an isomer. And an isomer is a concept of chemistry. It's very important. And essentially what an isomer is it is a molecule that has the same formula, but a different arrangement of those atoms. And there are multiple types of isomers. All right, there are multiple types of isomers. There are what we call cis and trans isomers. There are what we call constitutional isomers, and there are a very important category of molecules that are very relevant to us that are known as stereoisomers. Specifically, the category that I want to talk about today is what's known as an enantiomer. An enantiomer. And an enantiomer is a molecule that can be seen to have a right-handed form and a left-handed form. So these are molecules, the same chemical formula, but they are mirror images of one another. So imagine that you have your right hand, okay? And imagine that it is some molecule. What would the mirror image of my right hand be?
Your left hand.
My left hand, right? So I put my right hand in the mirror and I see my left hand, right? Now, are they exactly the same? Well, they have the same components, but everything's flipped, right? So I cannot superimpose, I cannot put my right hand on top of my left hand and make everything line up, right? They're non-superimposable. And that is how you want to view enantiomers. There are certain molecules, many molecules, that have a right-handed form to them and a left-handed form to them, okay? And these forms are mirror images of one another. And we often denote them as, if it is the right-handed form of the molecule, we call it the D. And if it is the left-hand form of the molecule, we call it the L. Sometimes you will also see R and S. R and S. And R is equivalent to D, and S is equivalent to L. All right. Now, the reason that you have this naming convention, the D stands for dextrorotary. And L stands for levorotary. And it has to do with the way that polarized light goes around these molecules. If you're talking about the D or the right-hand form molecule, when polarized light gets shined at that molecule, it will go around it in a clockwise. It'll go around the molecule. So here's the molecule in question, and I shine polarized light at it, and the polarized light will go around it in a clockwise fashion. That is what's known as dextrorotary, clockwise. Whereas if I have a left-handed form of the molecule, polarized light will go around that in a counterclockwise or an anti-clockwise direction. And that is what we call levorotary. So this is dextrorotary, and this is levorotary. And so when we look at glucose has two enantiomers. It has a D enantiomer and an L enantiomer. And the D enantiomer of glucose is dextrose. Does that make sense where that name comes from? So it is glucose, that it is the right-handed form of glucose. So it is a combination of dextrorotary and glucose, or D-glucose. Does that make sense?
Yes.
Okay, excellent. Now, interestingly enough, in nature, and we don't really know why this is, we don't really have a good understanding of why this is, but in nature, at least organisms like us, that we're aware of at least, we can only utilize the right-handed form of sugars for energy. So we can only utilize the right-handed form of glucose to make energy and do other biological functions, right? So we have to have that right-handed form. The left-handed form, the levorotary enantiomer of glucose, cannot be utilized by us. We don't know why that is, really, but the D form of carbohydrates are what we can use in nature. Now, interestingly enough, it's opposite when it comes to proteins. Proteins and amino acids, we can only use the L form or the S form, right? And so sometimes when you study amino acids, right, you might see like tryptophan, for example, is an amino acid that we use to make proteins and other molecules like neurotransmitters, like serotonin, right? So tryptophan. And if you're reading a textbook talking about tryptophan, you will often see an L in front of it, L-tryptophan, to denote that this is the levorotary form of tryptophan, which is the only form that we can use, right? So the D form of carbohydrates and the L form of proteins. Now, this becomes really relevant because in pharmacology, right, there are many drugs that have a right and a left-handed form to them. For example, glucose, right? And so we could not administer L-glucose to somebody and have the effects that we want. We'd have to administer D-glucose or dextrose. But there are some other examples of where this becomes important. And there is a term, something known as racemic. All right. And there are certain drugs that come in what's known as a racemic mixture. Racemic means a mixture of 50%. So 50% of the drug molecules are D, and 50% of the drug molecules are L. All right. So a racemic mixture is where you have 50-50, half and half, right? For example, a common drug, albuterol, you administer albuterol to somebody, which you all probably have, it's a very common drug. Albuterol is a racemic drug, and it comes in a mixture of D and L, right? So 50% of the albuterol you give somebody is D-albuterol, and 50% of it is L-albuterol. Now, there are other drugs where they're not racemic, and so there is a drug called Xopenex. And does anyone know what the generic name for Xopenex is?
Is it levobuterol?
Yeah, Xopenex is levoalbuterol, right? So it is a concentrated, right? You actually take the our form of albuterol, right? And you concentrate it. So you only take one of the isomers of albuterol, and that's what Xopenex is. It is a concentration of just one. It is no longer racemic. There are other drugs as well. For example, there's something called racemic epinephrine. And this is a kind of epinephrine that is often nebulized. We give it nebulized. Often we give it to children who have a supraglottic swelling. So croup, for example, right, where they have swelling above the glottis, and we can nebulize that epinephrine, and then that will agonize alpha receptors and help decrease the swelling. Sometimes we will give it to people who have been extubated. So we've taken an endotracheal tube out of them, and they have some swelling or some irritation, and we will give them some racemic epinephrine in order to decrease that supraglottic edema. So racemic epinephrine is another example where you actually have a racemic mixture of drug, 50% right-handed, 50% left-handed drug molecules. All right. So I just wanted to bring that up because that will, that is important pharmacologically and just biologically as well. All right. So now that we've gotten through that, what I want to do is I want to now move over and I want to redux some of the homework, quote unquote, assignments that I gave you all last week. And so now what I want to do is I want to talk a little bit about free radicals and reactive oxygen species. So let me ask you all a question. How would you define a free radical / ROS? And we'll talk about them as essentially being the same thing for today. But can anyone give me a working definition of a free radical reactive oxygen species?
The electrons are not paired. Is that correct?
Yeah, that is a fundamental idea, is that you have unpaired electrons So that's a critical concept, because electrons generally like to pair up. So electrons have a property known as spin. And some people, when you talk about spin, they think that they like literally spin. That's actually not the case. Spin is a purely quantum mechanical property, and it is just a I don't have any intuitive way of explaining what spin is, other than it's just a kind of a intrinsic kind of angular momentum that you have. And so when electrons go into these energy levels, there are what we call orbitals, which is kind of like a sub level of energy. And you can only fit 2 electrons in an orbital. And there is a very important principle that was developed when we started learning about this, called the Pauli exclusion principle. And essentially what it says is that certain kinds of particles, like electrons, cannot have the same state. It cannot exist in the exact same state, right? Some particles can, like light, like photons of light can. They can all be exactly the same and have the exact same state. But electrons, for example, cannot. They cannot exist in the exact same state. And so when we put these electrons, when these electrons inhabit these orbitals, you can only fit 2 in an orbital. And so what happens is the electrons have to have different spin. And when we talk about spin, we can either say that they have plus 1/2 spin or minus 1/2 spin. All right. Or often what we'll do is we'll just say, if we're talking about plus 1/2, that's spin up, and minus 1/2, that's spin down. All right. And so what happens is when you have a situation where you're putting electrons in an orbital, let's just draw an energy, right? So this is the energy on the y-axis here. So if you have an electrons coming in and you have an electron all by itself, maybe it's up here and spin up by itself. If I pair it up, these spins, the plus 1/2 and the minus 1/2 spins, kind of cancel each other out. And that actually lowers the overall energy, which makes it more stable, right? So when I have a spin up and a spin down, right? So the energy here is a little higher, the energy down here is a little lower. So that's a more stable configuration when you have a pair of electrons versus a single electron. And that higher energy in these reactive oxygen species, these free radicals, make them much more reactive. They're highly reactive because they want to do what? They want to get a hold of electrons. They want to hold electrons, and what they will do is they will take electrons from the molecules within our bodies, right? And so they can cause damage to DNA and damage to the cells. They can cause mutations and so on and so forth. So you have unpaired electrons, and you have unpaired electrons in these oxygen containing molecules. Oxygen is a very important part of this because oxygen by itself is highly reactive, right? It's highly reactive. In fact, oxygen is very toxic. So let me ask the next question. If oxygen is such a toxic, highly reactive molecule, why the hell is it so important? Why do we need oxygen if it's this toxic intracellular poison? Does anyone know why we actually need oxygen?
Is it to create energy?
Why though? What's so important about oxygen in energy production? You're not wrong. It's very important. And what is the primary molecule of energy that we use in biological systems? First of all, what is that? It's sometimes defined as the currency of energy in the body.
Is it ATP?
Yeah, ATP, right? Or adenosine triphosphate, ATP, right? High energy phosphate molecule. And then when that molecule dephosphorylates, right, it turns into ADP, adenosine diphosphate. So you can kind of imagine that maybe it's like a gun, if you will. And you've got the adenosine, and then you've got these PO4s, these phosphates. And when this last phosphate pops off, it flies off, kind of shoots off like a gun, if you will. And that energy that's released is what powers cell processes, right? And then it dephosphorylates and it becomes adenosine diphosphate there. But going back to the question, you're right that oxygen plays an important role in producing ATP, but where, why, and how? Does anyone happen to know what that's all about?
It's used for the aerobic metabolisms.
Yeah. So oxygen is important for ATP production, and specifically it's important for aerobic respiration. And what is that? What is that? Well, that is producing energy, ATP, in the presence of oxygen. So is there another way we can produce ATP?
Anaerobic metabolism.
Anaerobic, good. Anaerobic respiration, and that is producing energy without oxygen, right? Without O2. But anaerobic respiration produces very little ATP, not enough to power cells for the most part. There are some cells in our body that produce all of their ATP anaerobically, but these cells don't do anything. They don't even have a nucleus. They don't make proteins. And just out of curiosity, does anyone happen to know a cell in our body that can produce all of its energy anaerobically? Is there a cell in the human body that does not need oxygen to make energy? Because its energy requirements are very, very low. I'll give you all a hint. The cell doesn't have to do much. Its only job is to transport. That's all it does. It doesn't live very long. It doesn't make its own proteins. It just transports.
Red blood cell?
Blood cells?
Yep. RBCs, red blood cells. Good. Yep. Red blood cells. That's all they do. They float around and they have hemoglobin that transports oxygen. That's all they do. They don't do anything else really. And so their metabolic demands are so low that they don't need anaerobic aerobic respiration. Okay, so what I want to do is I want to spend a little time just talking about the major metabolic pathways as they pertain to energy production. So let's go ahead and let's do that right here. All right, so this is going to be an annotated view of what you all saw, hopefully what you read about. So these are some of the major metabolic pathways. So I'm going to start, and let me just choose a different color here. There we go. All right, I'm going to start here. I'm going to start up here. So I'm going to start outside of the cell, first of all. So this is extracellular. All right, and outside of the cell, we have glucose, right? And glucose, C6H12O6, can get into the cell. And so when glucose gets into the cell, it goes through a channel, through a protein channel, through something known as facilitated diffusion. And that's where insulin, right? Insulin attaches to the channel and facilitates the diffusion of glucose into the cell. And that's where this is going to start. So here I have glucose, right? These little dots are carbon atoms, 6 carbon atoms. And the very first cycle that glucose goes through is something called glycolysis. Now, we're not going to talk about every single step, but there are multiple steps. And at every step, there is an enzyme that catalyzes each specific reaction. So you can see this is a very big picture overview. And what the role of glycolysis primarily is to turn glucose into pyruvic acid. So glucose gets broken down into pyruvic acid. So you have this 6 carbon molecule glucose gets converted into, broken down into 2 3 carbon molecules, 2 molecules of pyruvic acid. Is everybody okay so far?
Yes.
Okay. So glycolysis is glucose gets converted into pyruvic acid, and in the process of doing that, a tiny amount of ATP is produced, like 4 molecules of ATP, but 2 molecules are actually used to power glycolysis. So you're netting like 2 molecules of ATP, tiny amount, right? Okay. Now, the next thing I want to do is I want to talk about the pyruvic acid. So pyruvic acid is an organic acid, and we said that most organic acids have a carboxyl group in them, right? A COOH. And when this hydrogen pops off, all right, so COO, okay, that's a minus. So that oxygen takes the electron, the hydrogen pops off as a hydrogen ion, all right? So when this hydrogen here pops off as a hydrogen ion, that's what makes these organic acids, right? This is a carboxyl group. Now, when organic acids do what acids do, their name changes, right? I think I mentioned this last week. So for example, we talked about formic acid. When formic acid gives off a proton, the leftover molecule, right? So this is the proton that popped off, and then this leftover molecule here gets its name changed, right? So the leftover molecule is a negatively charged molecule, and so formic acid becomes formate. And the ATE is to denote that formic acid did what acids do, right? Same thing with pyruvic acid. When pyruvic acid donates a proton, we call it pyruvate. All right, so pyruvate is just what happens when pyruvic acid does what pyruvic acid does, what organic acids do. All right. Now, everybody okay there? Everybody good so far?
Yes.
Okay, great. Now, pyruvate will get converted further into another molecule called acetyl coenzyme A, or acetyl-CoA. All right. If pyruvate does not get converted into acetyl-CoA and fed into another cycle, pyruvate will get converted into something called lactic acid. And under normal circumstances, that's not a huge deal, because the lactic acid will go into the liver and it will go into what's called the Cori cycle. The Cori cycle in the liver, right? And the Cori cycle essentially can turn the lactic acid, or rather the lactate, back into glucose as long as it's working okay. All right, everybody okay there? And this belongs to a larger process known as gluconeogenesis. Gluconeogenesis just means the production of new glucose, and this is one of the ways you can do it. All right. Again, this is if everything's working okay. So keep that in mind, because this will become a problem when the cell's not working. All right, so glycolysis is glucose gets converted into pyruvate. Pyruvate gets converted into acetyl-CoA, and then acetyl-CoA goes into the mitochondria here. So this yellow capsule is the mitochondria inside of the cells, right? And it will go into another cycle called the Krebs cycle. The Krebs cycle is sometimes referred to as the citric acid cycle or the tricarboxylic acid or TCA cycle. Those all mean the same thing, all right? And so these acetyl-CoAs are fed into the Krebs cycle. And what happens in the Krebs cycle is you have these carbons, right? These carbons are going into the Krebs cycle. And again, there are multiple steps, multiple enzymes going on here. We're simplifying this. But essentially what's happening is electrons and hydrogens are being tore off, are getting tore off of those carbons, right? And so what happens is the electrons and the hydrogens, electrons and hydrogen ions are being tore off, and they are getting attached to special transport molecules, specifically NAD plus and FAD plus. So as the electrons and hydrogen ions are being torn off of these carbons that are coming in from the acetyl-CoA, okay, the electrons and hydrogen are attached to these special transport molecules, NAD plus and FAD plus. And then NAD plus becomes NADH because it has taken on a hydrogen, excuse me. And then FAD plus becomes FADH2, right? So those are where the hydrogens and electrons go. And then they shuttle those hydrogens and electrons into another cycle known as electron transport. And then what happens is you have carbons and oxygens left over, right? So essentially you have, right? So coming in, you have carbons, oxygens, and hydrogens, right? The hydrogens get torn away. And so all you have left over are carbons and oxygens, and that produces CO2. So this is where carbon dioxide comes out as a waste. It comes out of the Krebs cycle as a waste. And then the hydrogens and electrons that were tore away from those oxygens and carbons are fed into the electron transport. And in the electron transport is where most of the adenosine triphosphate is produced. And the important thing to understand here is that oxygen, the reason we need oxygen to come in is we need some way of getting rid of those electrons and those hydrogens, those hydrogen ions, after they have been used to power electron transport. All right. So oxygen acts as an electron acceptor. It is the final electron acceptor in energy production. So oxygen comes in, it combines, oxygen comes in, it combines with the electrons and hydrogens to produce water molecules. So oxygen is kind of like an intracellular garbage truck. It comes in, it takes the waste that is coming out of electron transport and converts it into water. All right. So that's why we need oxygen. We need a highly reactive molecule that can accept hydrogens and electrons right? And if we don't have it, then we can't make energy. Unfortunately, it is highly reactive, it can be toxic, and it can form reactive oxygen species and free radicals. And so the very fact that our cells are functioning and making energy means that they're going to produce, under normal circumstances, normal day-to-day operation, we will be producing free radicals and reactive oxygen species all day long, right? And so we do have detoxification systems in our cells, but in many instances, pathophysiological instances, those detoxification systems can quit working, or they can become easily overwhelmed. All right. Are there any questions so far about the major basic pathways for producing energy: glycolysis, Krebs cycle, and electron transport? Is everything okay so far?
Yes, sir.
Okay. So what I'm going to do is, it's coming up on 9:30. Let's go ahead and take our first break. And then I'm going to talk a little more about how we can make energy with molecules, with other molecules, with molecules other than carbohydrates. So alternative energy production pathways. And then we'll start talking about what happens when all this starts failing. So I've got 9:25. So let's meet back up at 9:40. I will see you all back at 9:40.
See you there.
Yes, sir. Thank you. All right.
No, sir.
All right. Well, let me go ahead and pull the notes back up. There we are. Just kind of cleared that out to blank it out again. Okay, so in summary, glucose comes into the cell, and the first pathway is glycolysis, where glucose gets converted into pyruvate. A little bit of ATP is produced. And because glycolysis does not require oxygen, this is anaerobic respiration. All right. And then that pyruvate gets converted into acetyl-CoA. And then acetyl-CoA gets fed into Krebs cycle, where hydrogens and electrons are torn off of the acetyl-CoA and attached to NAD and FAD. And then NAD becomes NADH, and FAD becomes FADH2. And then that all gets shuttled to the electron transport. And then you have carbons and oxygens left over, and they pop out of the Krebs cycle as CO2, as waste. And the electron transport, these hydrogens and electrons that are being shuttled over there are used to power the electron transport and to create lots of ATP. And then once they have been utilized, the electrons and hydrogens have been utilized, oxygen comes in to accept them and make water molecules, right? So that's a big picture view of what's going on here. Now, let's say that for some reason, we are not able to make or get enough glucose in, or maybe glucose cannot get it into the cells, right? Maybe this is a type 1 diabetic, for example, is not making insulin. There are alternatives. There are alternative pathways for energy production. One of the important ones is using fats. Remember, we said that fats, the basic structure of fats in human are the triglycerides, where you have a glycerol head here, and then you have these three long chains of carbon capped with carboxylic acids, right? That is a triglyceride or a fatty acid. And what we can do is we can cut these long carbon chains up into little 2-carbon chunks or little 2-carbon segments, right? And so, and the way that we do that is through another metabolic pathway known as beta oxidation. And so essentially what you're doing is you're chopping off 2 carbons at a time. And you have these little 2-carbon molecules and acetyl-CoA. So you're chopping it down into little 2-carbon molecules and acetyl-CoA, and then that can be fed directly into the Krebs cycle, right? So this is how fats can be used as an energy source, right? By converting them into acetyl-CoA and other 2-carbon molecules in little intermediates that can be fed directly into the Krebs cycle. Unfortunately, this can produce lots of wastes. It can produce lots of organic acids, for example, right? Beta hydroxybutyric acid is one of the common ones that's produced, and it can produce ketones. And so what can happen is if you are breaking down lots of fat and producing lots of ketones and lots of organic acids, this can produce a disorder known as ketoacidosis. And this is where DKA comes from, right? Diabetic ketoacidosis, where somebody does not produce insulin, so glucose cannot get into the cells. So glucose levels increase substantially outside of the cells, and then the cells start breaking down fats to produce acetyl-CoA and other small carbon molecules that can be fed into Krebs cycle, so the cells can continue producing energy. But then this produces lots of organic acids and ketones as waste products, right? If you eat a diet incredibly high in fats and incredibly low in carbohydrates, you can do a similar thing. You can force your cells to begin breaking down fats, and that's called a ketogenic diet, right? And as long as you stay well hydrated and your kidneys are functioning, you can get rid of the wastes. And so you will still have ketones, you'll still have some ketones in your blood, but you won't be incredibly acidotic. And so some people just call this ketosis as opposed to ketoacidosis, right? And so there are some diets that can do this as well. In addition to that, we can also use amino acids to create energy. We can break proteins down into amino acids. And remember, amino acids is a molecule that has an amine group, an ammonia or a nitrogen containing group, a central carbon, and then a carboxylic acid group. And so essentially what we can do is if we can remove the amine group from these amino acids, we can make 2-carbon molecules that can be then fed directly into Krebs cycle. The problem with doing that is you create lots of ammonia, NH3, as a waste. Luckily, there is another cycle in the liver called the Urea cycle, where NH3 gets converted into a less toxic compound called urea, and then urea can get eliminated in your urine. So you can see the liver is incredibly important here. And when the liver begins failing, right, these various cycles that occur in the liver, like the Cori cycle and the urea cycle, begin to fail. And that's actually one of the major consequences of liver failure, is something known as hepatic encephalopathy. Does anyone know what encephalopathy means?
Is it tissue death?
Well, pathy is dysfunction, right? Enceph, does anyone know what enceph means? The prefix enceph.
Is that a type of swelling?
No. Think of the term cephalic. when we say that there is a cephalic presentation, right? If you're delivering a baby and there is cephalic presentation of the baby, what does that mean? Medical terminology is everything here.
Brain.
Brain or head. Yeah, head presenting. So inceph is a dysfunction of the brain that is due to the liver failing. And the reason that happens is the urea cycle in the liver quits working, and ammonia cannot be converted to urea. And so what happens is ammonia levels increase in the body, and the ammonia, it is the ammonia that causes the brain to begin shutting down. And so these people have confusion, behavioral changes, and so on due to that hepatic dysfunction, right? So again, we're just making all sorts of different connections here. All right. So that's kind of the big picture of some of these major metabolic pathways and a little bit of pathology associated with them. So let's go back now to the reactive oxygen species and free radicals that we were talking about a little earlier. And let's do a deeper dive. So again, what are they? Well, they're oxygen-based radicals that have unpaired electrons in their valence, and they're highly reactive, like we talked about earlier. And so here are some examples, right? So if I have just molecular oxygen, right? Molecular oxygen is fairly stable. It's fairly stable. Its octet rule is met, right? It's 2 oxygen atoms, double covalently bound. So here are the covalent bonds in the middle here. Let me get rid of. There we go. So I'll circle those, right? And then you can see pairs of electrons on each oxygen atom, fairly stable. What happens is, if this oxygen molecule were to pick up an extra electron here, I'll circle that extra electron, that's an unpaired electron. And now that oxygen molecule has become highly reactive because that electron wants to go somewhere. And so this is what's known as a superoxide anion. It is a free radical version of oxygen, if you will. And remember, in energy production, we said a lot of it is electrons and hydrogen ions are getting shuttled around, right? So the risk of an electron bumping into oxygen and creating a superoxide anion is going to be elevated in cell respiration. Hydroxyl as well. So here you have a hydroxyl ion, right? Remember, we talked about hydroxyl or hydroxide groups or alcohol groups. So this is just an alcohol. This is just an alcohol ion, but you can see that all the electrons are paired up, and you've got that covalent bond with the hydrogen, right? So that's fairly stable. But again, if an electron gets transferred to that oxygen on the hydroxyl ion, you now have an unpaired electron, and it wants to go somewhere. It wants to interact with something. It's highly reactive, and it's a higher energy. And this is what we call a hydroxyl radical, right? So those are just 2 specific examples of these reactive oxygen species or these free radicals. So the next question is, where do these come from? Well, by and large, reactive oxygen species and free radicals come from normal cell respiration, right? Just normal energy production. Like I said, oxygen is a toxin. And so most of these free radicals actually come out of the electron transport chain. So what I want to do is I want to spend a little bit of time talking about the electron transport chain. So I'm just going to zoom in here a little bit. All right. And let me move that over so you can all see that. Okay. So what we're doing here is we're actually taking a look at the electron transport chain inside of the mitochondria. So right here is glycolysis in the Krebs cycle. And remember, we said coming out of the Krebs cycle, you have hydrogens and electrons being attached to NAD and FADs, right? They're the shuttles. And so the NADs are NADHs and the FADs are FADH2s. And they are coming in, and what they are doing is they are transferring hydrogen ions and electrons into the electron transport. And the electron transport is a chain of multiple proteins, right? Called cytochromes and complexes. And so here on the left is complex 1, and then complex 2, complex 3, cytochrome C, and complex 4. Collectively, these are known as cytochrome C oxidase, because this is where oxygen is used. And then at the very end is where all the ATP is produced, and this is a protein. So all the blues, the blue colored things that are going through the membrane of the mitochondria are the proteins that power the electron transport. And on the very end, there's a special protein called ATP synthase. And this is, as the name suggests, where most of the ATP is synthesized. All right. So the NADHs come in and the FADH2s come in, and they drop off the hydrogen ions and the electrons. All right. The hydrogen ions go up here, and they are collected in a big reservoir. So think of this as like a big reservoir, or you could draw this a little differently where I have a dam. And then I've got a little pipe that goes through that dam, we'll say. All right, so something like this. And then I've got lots of water here. All right. So think of the hydrogens as collecting way up here. You got a big collection of hydrogens. And then those hydrogens are going to pass through this pipe in the dam, and they're going to spill out down below, right? And that pipe, where the hydrogens will go through, right? So if this is a dam, you could put a generator in there, right? You could put a turbine and you could generate energy as that water passes through that pipe. It spins a turbine around, and that's what we refer to as hydroelectric power. Well, something very similar happens here. These hydrogens accumulate up here, and then the hydrogens will pass through this channel here called ATP synthase. And as the hydrogens pass through the channel, they literally cause it to turn around, kind of like a propeller. It's actually something referred to as proton motive force, or PMF, proton motive force. It literally spins a channel around. And as that channel spins around, what it does is it can combine phosphate and ADP. So ADP combines with phosphate, and lots of ATP molecules are spitting out of here. All right. Now, the energy from the electrons, because remember, electrons are also being deposited here, not only hydrogens. The energy from electrons are being deposited, and that energy from the electrons power these protein complexes that allow for that buildup of hydrogen, or protons, hydrogen ions. Okay, so you can see these electrons are coming in here at high energy. They're cascading through these protein complexes, and then they come out down here at low energy. So what you have left over coming out, you have a bunch of hydrogen ions spitting out of here, and you have a bunch of electrons, low energy electrons spitting out, and we need some way of getting rid of them, right? And again, this is where oxygen comes in. So O2 comes in, O2 is split apart into just oxygen atoms, very highly reactive oxygen atoms, right? And that oxygen, okay, can combine with 2 hydrogen ions and 2 electrons. So 2 hydrogen ions, 2 electrons, and 1 oxygen atom combines to form H2O. And then that takes that waste away. So more hydrogen ions and electrons can be transferred into here, and more ATP can be produced. So what happens if the oxygen doesn't come in? Well, if oxygen doesn't come in, then it can't act as a receptor. Hydrogen ions back up here. That shuts this down. You can't produce ATP anymore. Electrons back up. That shuts this down. This backs up. It shuts down. It backs up into glycolysis, or rather the Krebs cycle. The Krebs cycle shuts down. And then going back here, okay, so electron transport system has shut down, all right. Krebs cycle has functionally shut down. And the only cycle that continues to work because it doesn't need oxygen is glycolysis, right? So glycolysis continues to work, but it can't produce enough energy to power the cell, right? So it just keeps pumping out pyruvic acid, which gets turned into pyruvate. But what happens to pyruvate if it does not go into the Krebs cycle? Do you all remember what happens to it when it does not go into the Krebs cycle?
The Cori cycle.
Right. But we just said that there isn't enough oxygen. So is the Cori cycle in the liver going to work if we're not getting oxygen to the cells? What do you think?
Negative.
No. So the Cori cycle is not working very well. And what does pyruvate get turned into?
Pyruvic acid.
Yeah, it gets turned into lactic acid, right? And lactic acid would normally go into the Cori cycle, but we're talking about an anaerobic environment right now, and the Cori cycle isn't working very well. So what happens is you get an accumulation of lactic acid in the cell. So a couple things happen. You have a decrease in ATP, and the metabolic pathways in the cells quit working. The cells cannot make energy anymore. In addition to that, you have a buildup of lactic acid, right? So you have acidosis, right? And the various ion pumps that pump ions in and out of the cells fail, all right? And then the cells cannot regulate water going into and out of them, right? Because water goes into and out of through little pores called aquaporins, and that's modulated by ion concentrations. And so what happens is water starts coming into the cells, and the cells begin to swell. So the cells quit working, they begin to swell, and they become very acidotic. And this is what we see in sustained shock states, right? This is what we see in sustained shock states when you have cellular hypoperfusion. These are the metabolic cellular level changes that we see occurring, right? And in fact, lactate levels, right? Because lactic acid becomes lactate when it gives off hydrogen ions. We can measure that, and that is sometimes used to see how somebody in shock is, how they are responding to therapy, right? If their lactate levels are continuing to increase, that means that they are continuing to have hypoperfusion, whereas if lactate levels are starting to go down, then that means that the patient is improving and that their perfusion status is getting better, and that, right? So are there any questions over that, or does that make sense so far?
No question.
All right. Okay, so going back here. All right, excellent. So another way that we get lots of free radicals is in shock states. If we get free radicals in normal cell respiration, we're going to get more free radical and reactive oxygen species when there is abnormal cell respiration, right? And to make it even worse, we tend to get increased reactive oxygen species when ischemia is reversed. So when somebody has been in a state of hypoperfusion for a long period of time, if we reverse that, what happens? Well, now the cells are getting perfused and you've got lots of oxygen coming back into the cells, but the cells' metabolic pathways are not working well. And so what happens is you can get lots of reactive oxygen species, and this can produce something known as a reperfusion injury. And we sometimes see this in patients who have had heart attacks or acute myocardial infarctions or strokes, where when we reverse the pathology, we can get some additional damage occurring due to the reperfusion, right? And so sometimes people that are, for example, having a heart attack or an acute myocardial infarction, when we open their blood vessel back up and blood begins reperfusing the heart, sometimes the heart will develop dysrhythmias. The heart will become very irritable for a certain period of time, and there may be some increased injury due to the reperfusion. Additionally, if you have abnormal fat or lipid metabolism, you can create lots of reactive oxygen species, right? And so this is where we talk about various disorders of lipid and fat metabolism, right? You can have dysfunction. You have a dysfunction and lots of reactive oxygen species develop. For example, there's a disorder called steatosis. I think I mentioned this last week. One of the major organs where we see this is in the liver. This is sometimes referred to as fatty liver disease or non-alcoholic fatty liver disease, NAFLD, or non-alcoholic fatty liver, right? So this is where the liver, normal hepatocytes begin to be replaced with fat, and that is very inflammatory and produces lots of reactive oxygen species, and then that can go on to actually cause liver failure, cirrhosis, and other problems, right? And so people that have abnormal fat or lipid metabolism, morbid obesity and obesity, for example, right? You can create lots of reactive oxygen species. Sustained inflammation, and we're going to talk about inflammation when we talk about immunopathology. But inflammation is kind of one of the first things that happens with your immune system, right? If you develop an infection or you have an injury, one of the first things that happens is inflammation. It's part of the innate immune system. And you have phagocytic cells. Does anyone know what phagocyte means, or what does it mean to phagocytize? What does that mean?
It's a process called phagocytosis where the phagocytes, they pick up the antigen or like cellular debris.
Yep, it literally means to eat. Yeah. So these cells are floating around, and then you have some antigen or some cellular debris, and they come in and they literally kind of engulf it and eat it. Yeah, that's phagocytosis. And there are several phagocytic cells, for example, neutrophils and macrophage, as well as dendritic cells, right? These are all phagocytic. Phagocytic cells produce lots of reactive oxygen species, because what they can do is they can release those highly reactive molecules to help denature and destroy those antigens and make them easier to phagocytize. So if you have sustained inflammation, so inflammation that doesn't go away or too much inflammation, you will have increased production of these reactive oxygen species. And there are whole disorders of sustained inflammation, right? We sometimes refer to these as hypersensitivity reactions and autoimmune disorders, and we will talk about some of them in some detail. But this is the cellular pathophysiology that we see going on here. And you also have lots of different toxins that can produce reactive oxygen species. For example, acetaminophen is a classic one. What happens with acetaminophen overdose? What organ is primarily affected or impacted?
The liver.
The liver, yeah. So what happens is one of the important molecules involved in detoxifying reactive oxygen species. So we have lots of different molecules in our bodies, systems to help detoxify reactive oxygen species. These are sometimes referred to as antioxidants. Right? And one of the important antioxidant molecules in our body is something called glutathione. And what happens is if you have too much acetaminophen, some of that acetaminophen will get converted into a very toxic radical called NAPQI. And if you have too much NAPQI, it overwhelms the glutathione. And so you have a depletion of glutathione. And now you have these reactive species are developing, but there is no antioxidant to deactivate them. And then they begin damaging and destroying cells and tissues where they're being produced. And this is primarily in the liver, because the liver is where drugs like acetaminophen are metabolized, right? And so you see this primarily occur in the liver, right? We also see this with iron. If somebody overdoses on iron, this can produce lots of reactive oxygen species, can be very toxic, specifically to the gastrointestinal tract, right? So someone overdoses on like iron supplement, for example. All right. So those are just some examples of where we see reactive oxygen species being developed. And this is collectively often referred to as oxidative stress. So any situation where you have an abnormal increase in reactive oxygen species and free radical development is known as oxidative stress. So if you hear somebody say the cells are under increased oxidative stress, you know that whatever is causing that, it is causing increased reactive oxygen species and free radicals. All right. Okay. So any questions over that? Any questions over that?
No.
Does that make sense? Where oxygen is important, where it's used, and more importantly, is everybody okay with some of the major things that happen when there is cellular hypoperfusion? Everybody okay with that? Okay, because I know that was, we went over a lot of information and we went into a good amount of detail. So I just want to make sure you're all comfortable with the concepts I covered there. Okay, well, excellent. So now, let me stop here. All right, pull my video back up. All right, excellent. So now what I'm going to do is I'm going to move on and we're going to begin talking about acid-base balance and how we assess it, how we analyze it, and some of the major problems that we can have. And this does dovetail fairly nicely into what we've been talking about so far. Again, this is going to be quite a bit of information. And a little bit of math too. So I just want to make sure you're all comfortable with everything that we've talked about so far. Everybody okay so far? Okay, so what I'm going to do is I'm just going to do like a introduction to acid-base imbalances, and then we're going to get into the details and hopefully get into talking about the various disorders of acid-base a little later on today. All right, so let me go into screen share here. I'm sure you all can see. Shut my video off so the audio is hopefully a little better. All right, here we are. Acid-base imbalance. So why is it important to understand acid-base imbalance? Well, it's important to understand acid-base imbalance because multiple pathological processes can cause acid-base abnormalities or anomalies, as we just talked about, right? Just hypoperfusion can cause, right? Just being in shock and having hypoperfusion can cause organ failure, can cause renal and pulmonary problems, which manifest as acid-base imbalances. Even certain overdoses can cause acid-base imbalances. So these are important to understand, right? And it's important to also know that not only does acid-base imbalance, right? So if you have an acid-base abnormality, right, the alteration in pH itself can cause problems, right? So when you have a pH alteration, not only do you have whatever's causing the pH alteration, but the too low or too high pH itself can impact homeostasis, right? More importantly, pH changes can denature proteins, right? We talked about the protein conformation last week, and we said that the shape of a protein determines its function, and if the pH changes in a way that causes an abnormal shape to form, that protein may weaken what we know it, right? And that can be different enzymes, that can be clotting factors, right? So different enzymes and enzyme systems can quit working or work abnormally, clotting factors, right? Ion pumps and ion channels, different cell receptors, right? These are all proteins, right? So this is very important, right? Hemoglobin, we talked about hemoglobin can begin to function abnormally if the pH is too high or too low, all right? So that's the motivation for having to understand this in some detail. So when it comes to talking about acids, there are two major kinds of acids in the body. They're what we call volatile acids. And what does volatile mean? When you hear the term volatile, what does that mean to you? What do you all think? We say something unstable. Unstable. I think that's what most people would think, right? that something volatile is unstable. Actually, what volatile means is how easily something becomes a gas. How easily does it turn into a gas or vapor? So when we say that gasoline is volatile, we don't mean that it is explosive. Well, it is explosive, but that if you spill gasoline on the road, it evaporates very rapidly, right? So gasoline does not it does not remain a liquid for long because it evaporates very rapidly. That's actually why we say gasoline is volatile. So when we talk about volatile acids, these are acids that can be eliminated easily via the respiratory system. And this is what I want you all to know. There's really only one volatile acid. It's an important one, though, and that is carbon dioxide, CO2. But wait a minute. Let me ask you all a question. What is an acid again?
Anything that can donate a hydrogen.
Anything that dissolved in water donates hydrogen ions, specifically. So if we look at carbon dioxide as a carbon double covalently bound to oxygens, if we look at that molecule, does it look like it has any hydrogen ions to donate? CO2, what do you think?
Well, it doesn't, but if it's mixed in the blood, then it's formed into a carbonic acid, right?
Oh, look at that. You're stealing my thunder. Yes. So carbon dioxide by itself is not an acid, but carbon dioxide is a non-polar molecule, right? It's non-polar, which means, does it easily dissolve in blood? What do you all think?
No.
No, right? Non-polar molecules do not dissolve in polar liquids. And remember, our blood is primarily made of water, and water is a polar molecule. So what happens is carbon dioxide has to get converted into something that can dissolve in the blood. And so as carbon dioxide is pouring out of Krebs cycle, what happens is CO2 is combined with water. And there's an enzyme called carbonic anhydrase that catalyzes that reaction. So CO2 is coming out of the cells, it chemically combines with water, and it forms a new molecule called H2CO3. That's also known as carbonic acid. And then carbonic acid does what acids do. Some of the carbonic acid molecules, because it is a weak acid, so not all of them, but some of the carbonic acid molecules break apart into hydrogen ions and bicarbonate ions. And because hydrogen ions, bicarbonate ions are charged, they can dissolve in water. So they will dissolve in the blood, and then they will get transported to the lungs, where in the lungs, they will recombine in the presence of carbonic anhydrase. They will recombine into H2CO3, and then the H2CO3 can break down into CO2 and H2O, and the carbon dioxide can get eliminated in the lungs. So this part of the chemical equation happens in the tissues. All right. This part here is in the blood. And then this part here happens in the lungs. specifically in the alveoli, right? And as long as carbon dioxide is coming out of the tissues, it will push the chemical reaction in this direction. But sometimes, right, you can have things that can push the chemical reaction in the other direction, because remember, I drew these arrows in both directions. So this is an equilibrium reaction. It is a reversible chemical reaction, so it can get pushed in multiple directions depending on what's going on. But this is what I want you all to know, at least at this point. CO2 combines with H2O to make H2CO3, or that breaks down to hydrogen ions and bicarbonate ions, get transported to the lungs, where they recombine to form CO2 and H2O, and the carbon dioxide gets exhaled off, right? So it is the fact that the carbonic acid is releasing hydrogen ions that makes it an acid, all right? So that is the only volatile acid we have. And then there are what we call fixed acids. And fixed acids are acids that cannot be eliminated through the lungs, and they typically need to be eliminated through the kidneys. They require renal clearance. So it should come as no surprise that one of the complications of renal failure is an inability to regulate acid-base balance, right? Acidosis tends to be very common in renal failure because the body cannot get rid of fixed acids. Now, with that in mind, before we take our next break, I also want you to be aware that there are what we call buffer systems. A buffer system is a physiologic system in the body that prevents sudden changes in pH. So if something happens in your body where you start making a lot of acid, right? So let's say that maybe you develop some hypoperfusion of some cells in your body. These buffer systems prevent the pH in the body from changing, right? So they recognize, oh, look at that. We got lots of hydrogen ions being produced here. Let's prevent the pH from changing suddenly. And there are three major buffer systems that we need to be aware of. There are more than these, but these are the three major ones. All right. The first one is what's called the blood buffer system. This is the most rapidly acting buffer system. It acts within seconds, but it is easily overwhelmed. And the way that this works is, and this ratio is going to be important, so I want you to remember this. It'll be important for some of the math that's going to come a little later on. You have a ratio of carbonate ions to carbonic acid. You have a 20 bicarbonate ions, and remember, bicarbonate is a base, and bicarbonate is the major base, right? Bases accept hydrogen ions. So you have 20 molecules of bicarbonate ions to every 1 molecule of carbonic acid in your body. And remember, carbonic acid comes from CO2. So 20 to 1. And when that ratio begins to decrease, right? So maybe it's 15 to 1. That means that some of those bicarbonate ions are being used to buffer acids that are being produced. 10 to 1, 5 to 1. And as that ratio gets smaller and smaller and more of that bicarbonate gets used up, the pH will then begin to change, and the buffer system will no longer be able to prevent sudden pH changes. Now, the next buffer system is the respiratory system. And the respiratory system acts within minutes to hours. And the way that it works is it can either increase or decrease minute ventilation, or what we call VE. That's the amount of air that goes in and out of your lungs per minute. And that is the respiratory rate multiplied by the tidal volume, or the VT. And essentially what you're doing is you are blowing off CO2, and the lungs will blow off CO2 if the body is getting acidotic, or you will hold on to CO2. You will retain CO2 if you are getting too alkalotic. So you will breathe faster and deeper if you're getting acidotic, and you will breathe slower and shallower if you're getting too alkalotic. And then finally, the last and the definitive buffer system for the kidney, the renal buffer system. However, this takes hours to days. So this is a very slow one, right? And it can either hold on to or eliminate hydrogen ions and bicarbonate ions. And then you have some other very minor, you have proteins in your blood, like albumin, and you have phosphate that act as very minor buffer systems. But these are the three major buffer systems: the blood buffer, it's the rapidly acting one, but it's easily overwhelmed; the respiratory buffer, it takes minutes to hours to begin to compensate; and then the renal, and it takes hours to days. All right, any questions over any of that before we go into our next break? Is everybody okay there? So
I'm good.
Good.
awesome. All right. And I know we're probably going in, at least for many of you, we're probably going into a kind of detail that maybe you've never encountered in your prior courses. So I want to make sure everybody's okay so far, that we're getting things. Okay, chat looks okay. All right, well, it's 10:30. Let's see, 10:45. I will see you all back at 10:45. All right, so before we get going, are there any questions? Anybody have any questions over anything?
No, sir.
All right, well, let me go ahead and I'm going to stop my video just so I can hopefully help with sound. All right, so let's go talk about acid-base imbalance. So the thing that we need to understand first is, how do we determine acid-base in a human being? And we talked a little bit about that last week, the chemistry of life. So just a quick review here. Remember, we said in the 1800s, approximately 1880, we said Arrhenius came around. And said that essentially hydrogen ions have something to do with acid levels. And then in the 1920s, right around 1923, Bronsted and Lowry came along. And they said acids are anything that donate protons when it dissolved in water, and bases are anything except protons. And then in between there are a couple of people. In 1908, Henderson, a guy by the name of Henderson, and then in 1917, another guy by the name of Hasselbalch developed an equation that relates different components of acid base, and this has since been known as the Henderson-Hasselbalch equation. And then finally, by about the 1950s, this is where physicians begin to realize, hey, we can begin to use these concepts that were developed, specifically Bronsted-Lowry and Henderson-Hasselbalch, and we can begin using this in the context of human physiology. And that's what we're going to kind of do here, is we're going to kind of recreate what they did and recreate the mathematical basis, the quantitative basis for understanding acid-base balance, at least for our purposes. And so what they and what we should realize at this point is that pH, right? pH of the human body, the power of hydrogen, right? Remember, pH is just the negative log of the hydrogen ion concentration, is primarily a function of two things in the human body. All right, so pH is mainly a function of two major things. It is a function of the PaCO2. And whenever you see a little a in physiology always means artery. So this is the pressure, the partial pressure of dissolved carbon dioxide in the arteries. That's what that means, PaCO2. Big A means alveoli. Little a means artery. So pH is mainly a function of PaCO2, right? How much carbon dioxide is dissolved in our blood, and how much bicarbonate do I have dissolved? HCO3 negative. That's mainly what pH is a function of. So pH is the result of an interplay between CO2 and bicarbonate, right? And they realized that they could bear or bring some mathematical rigor to this, all right? And so first of all, what I want to do is I want to revisit the concept of a weak acid. A weak acid. And remember, we said a weak acid is an acid that does not completely dissociate. So something like hydrofluoric acid or HF, if you put hydrofluoric acid into water, 100% of the hydrofluoric acid molecules will dissociate into hydrogen ions and fluoride ions, right? Now, if we take a weak acid like carbonic acid, and I dissolve that in water, not every molecule will dissociate. Some molecules will dissociate into hydrogen ions and bicarbonate ions, but some of those molecules will not. They will remain carbonic acid, right? And so that makes this a reversible reaction versus the irreversible reaction we see with a strong acid. So because we're talking about CO2, CO2 is a weak acid, right? And there is a way that we can figure out how much something will dissociate into an acid. All right. And this is something referred to as the dissociation constant. All right. But prior to that, another way that we can look at this is in the following. Some generic weak acid we can just symbolize as being HA. HA is just a generic, could be carbonic acid, could be some other lactic acid, right? So it is just a generic, is a symbol. It is a symbol for some generic acid. And when that dissolves in water, all right, whoops, let me, so when that dissolves in water, all right, some number of those will dissociate, and they will dissociate into hydrogen ions, and then whatever is left over. So when a generic acid gets rid of its hydrogen ion, whatever is left over is a negatively charged anion, or A minus. And I'll give you an example here in just a minute. All right. Now, Henderson-Hasselbalch say that some generic acid will dissociate into its acid form, and they call that the conjugate acid, and its non-acid form, and this is the conjugate base. All right. And oftentimes what we will do is we will put brackets around those, like so. And in human physiology, whenever you see a bracket, if I put a bracket around something, some value X, that bracket just means that I am talking about the molar concentration, right? In moles per liter. That's all that means. All right. So let me give you an example. Let's say that I have, let's see, what do I want to do? Well, let's take lactic acid, for example. Lactic acid. Lactic acid has, this is the chemical structure of it. And every little line is just a carbon. So I'm just going to kind of simplify this a little bit. Oh, stop. OH. And then this is a CO. OH. There's a carbon here, carbon here. Yeah, I guess I'll just draw it. I'll just draw it out, just so we're not being too confusing. Carbon there, there's a carbon there, there's an OH, there's a carbon OH. All right, so this is lactic acid. All right. So this is our generic acid. We would call this the HA. All right. This is lactic acid. So this is HA. So when lactic acid gets dissolved in water, right? All right. So lactic acid plus water. All right. Some number of lactic acid molecules are going to dissociate. All right. And so they're going to dissociate into, so what's going to happen is this hydrogen here is going to pop off as a hydrogen ion. Remember, this is a carboxyl group. This is what makes lactic acid acidic, this carboxylic acid group here. All right. And so what you're going to get is when that dissociates, it is going to dissociate into a hydrogen ion. And then what's left over after that hydrogen pops off is the following. So you still have this, so you still have the CH3 here, the methyl group, the carbon, the OH, and then the COO. And this is going to be negatively charged because that oxygen took that electron with it, right? So this is the acid, the conjugate acid, and then this is the leftover part, the negatively charged leftover, the conjugate base, right? So this is lactic acid here, and then it dissolved in water, dissociated in hydrogen ions, and you're left with hydrogen ions and a lactate molecule, like so. All right, so that's how it works. So what we can now do, going back, is we can figure out the dissociation constant for any weak acid. And that's just a way of figuring out how much of it is going to dissociate. And the dissociation constant is something we just have to experiment to figure out, is also known as the Ka. So whenever you see Ka, that just means dissociation constant. And the way that we figure that out is we take a solution, all right? In the case of human physiology, we're going to be working with carbonic acid, but, right? You figure out the molar concentration of hydrogen ions to the concentration of A minus. All right, so remember, your weak acid, your HA, when it dissolves in H2O, will dissociate into hydrogen ions and anions. All right, so the conjugate acid and the conjugate base form. All right, so what we do is we measure, okay, what's the hydrogen ion concentration and what are the concentration of conjugate base, and then we divide that by the concentration of the acid that is undissociated, the HA, right? So when you put, right, you have some known concentration, right? So you put, so many moles, right? If I put like 1 mole of some acid, some weak acid into 1 liter, right, of water, so I have 1 mole per liter, and then I mix it up, and then I measure, and let's say that this is, the H plus is 0.5 moles per liter, and the A minus is 0.5 moles per liter, right? When I mix it up. So this would be 0.5 times 0.5 divided by 1, right? 1 mole per liter. And then 0.5 multiplied by 0.5 divided by 1 would be the Ka, the dissociation constant for whatever acid we're talking about. Okay, that's how we calculate the Ka. That's where it comes from. Now, what we tend to do is we don't use the Ka in physiology, but rather what we do is we will take the negative log of the Ka value, because we're talking about pH ultimately. And so if I take the negative log of the Ka, that equals a new concept known as the pKa. The pKa is just the negative log base 10. We're always going to be talking about base 10 here of the Ka. All right. And mathematically, what this comes out to be is the following. The pKa equals the pH plus the log of A minus, the conjugate acid, divided by the undissociated concentration of acid HA. All right, so that's a formula that comes out of that. It's not the formula that we want, we're getting there, but that's a formula that comes out of that. So what we can do is we can rearrange this formula, because what we're most interested in is we want to be able to solve for the pH, right? Because back up here, right, when we started this is what we're talking about. Hey, how do we get to the pH in a human being? All right, so what I need to do is I need to take this formula and I'm going to rearrange it to solve for the pH. So I want pH to equal, right? So I'm going to rearrange that formula, pH, the pH on this side equals, all right? So pH is going to equal, and then it's going to be pKa, right? That's just the log of the Ka, the negative log of the Ka. pH equals pKa, right? Hopefully you can see where that kind of came from. Plus the log, all right? A minus in molar divided by HA. And this is the equation that comes out of that. pH equals the pKa plus the log of A minus over HA. So remember, A minus is the conjugate base, and HA is the undissociated acid in its undissociated form. And this is actually known as the Henderson-Hasselbalch formula. All right, so this is the main formula of acid-base balance, at least as it pertains to human physiology. All right, so let's go back and let's now make this formula relevant to the human. Okay, so pH, well, that's relevant. So here's the question I want to ask is, well, what is the major weak acid in the human body? What do you all think? What's the most relevant weak acid in the human body?
Lactic acid.
pH is a relationship. No. So remember, pH, the major relationship is the following, right? Let me draw that.
Bicarbonate acid?
Carbonic.
Well, bicarbonate's not an acid, though, right? It's a base.
Carbonic acid?
Yeah, right. So CO2 is the major volatile acid, and it gets converted to carbonic acid. So what we want to do is we want to figure out what the Ka is for carbonic acid. That's H2CO3. And more specifically, what we want to do is we want to figure out what the pKa of carbonic acid is. And I'm just going to give it to you all. We don't need to calculate it. So what I'm going to do is, as we've already figured it out, and we know that the pKa, so here's the pH, the pKa of carbonic acid is 6.1. So pH equals 6.1, right? Plus the log, all right? And then let's go ahead and do this. So what is the major conjugate base when talking about carbonic acid? So H2CO3. When it breaks down into hydrogen ions.
Bicarbonate.
Yeah, HCO3 is the major conjugate base. So the A minus is going to be the bicarbonate concentration in someone's blood, right? The A minus there. And then the HA, the undissociated acid, is going to be what? Well, it is going to be the carbon dioxide, right? That is the OG form, right? It comes out as carbon dioxide. So it is going to be the PaCO2. But then what we need to do is, because not very much carbon dioxide dissolves in your blood because it's nonpolar, we need to take that into consideration. And there is a solubility constant, and there's actually a gas law. It's referred to as Henry's law of solubility. And so we need to apply Henry's law to that. And I'm just going to give you the number. It is 0.03. So CO2 times 0.03, all right, is the HA. That is essentially converts PaCO2 into, all right, into H2CO3. That's how much of it will get converted into carbonic acid. So that would be the HA, or yeah, HA, the undissociated form. All right. Now the next thing is, what we'll do is we will take hundreds of thousands of people who are well. So we'll take lots and lots of people who are normal. Lots and lots of normal people, and we will measure the bicarbonate, okay, the HCO3. We will measure the bicarbonate concentration in their blood, and we will measure the PaCO2 value in their blood, because we want to figure out what is normal, right? What are normal values? And when we do that in tens, hundreds of thousands of people, we find that the normal value for bicarbonate is 24 milliequivalents per liter, right? And the perfectly normal PaCO2 is 40 millimeters of mercury. That's the partial pressure. This is all pressure, right? So what we're going to do now is we're going to plug those values into the Henderson-Hasselbalch equation. All right. So HCO3 is 40 milliequivalents per liter. And because it's just a negative one charge, it's millimoles per liter. So it is already in molar concentration. And then the normal CO2, oops, I said 40, didn't I? 24, excuse me, it's 24. 24 milliequivalents per liter, so 24. And then normal CO2 is 40. So I'm going to rewrite this as the normal pH equals 6.1 plus the log of 24 divided by 40 multiplied by 0.03. Right. And now what I'm going to do is I'm going to begin to solve this. All right. I'm going to begin to solve this equation. And so the first thing I'm going to do is I'm going to multiply 40 multiplied by 0.03. So I'm going to do that first. pH equals 6.1 plus the log of 24 divided by, and 40 multiplied by 0.03 gives me 1.2. All right. And then I will divide 24 by 1.2, and that gives me pH equals 6.1 plus the log of 24 divided by 1.2 gives me 20. The log of 20. Huh. That's interesting. So that gives me 24 divided by 1.2 gives me a 20 to 1, right? It's 24 divided by 1.2 is 20, right? That gives me a 20 to 1. Is there anywhere where 20 to 1 has been talked about today?
Bicarbonate ratio.
Ah, the bicarbonate to carbonic acid ratio. When I talked about the bicarbonate buffer system, isn't that amazing? It literally falls out. The 20 to 1, the normal bicarbonate, right? This is the bicarb to carbonic acid ratio literally falls out of the Henderson-Hasselbalch equation as we are solving it. All right. It's astonishing. It's actually kind of amazing. All right, well, let's continue to solve this. Oops. There. All right. So pH 6.1 plus the log of 20. So now what I'm going to do is I'm just going to take the log base 10 of 20. All right. So the log of 20 is 1.3. So pH equals 6.1 plus 1.3. And what does that give me? What's 6.1 plus 1.3?
7.4.
Say that again.
7.4.
You got it. 7.4. And this is in fact the normal pH that we see. So if somebody has a bicarbonate of 24, a PaCO2 of 40, they will have a pH of 7.4. And that is the interrelationship there. Talked about there. So this is a quantitative basis for acid-base balance. And so when we talk about assessing the acid-base balance in a person, it should come as no surprise that those are the three values that we are going to look at. And the way that we determine acid-base balance is something called an ABG, and that stands for arterial blood gas. We have a sample of arterial blood, and then we analyze it. And what we do is when we analyze it, we get four major values that we're going to be looking at from an ABG. The first is a pH. You'll get a pH. The second is the PaCO2. The third is the bicarbonate, the HCO3 minus, and then the fourth is the PaO2. All right, and those four values are going to tell us most of what we need to know. So the pH is pretty self-explanatory. That's the pH of the arterial blood. All right. What is normal pH again? What is normal pH?
7.345 to 7.45.
No. Get that out of your head now. What is normal pH?
7.4.
7.4 is normal. All right. So 7.4 is normal. All right. That is perfectly normal. Now, there is an allowed range, but this is how I want you to look at that range. Remember, lower pHs equals more acid. So 7.35 to 7.39. So when looking at pH, 7.35 to 7.39 is what I call acid normal. It is normal, but it is in the acidic range. 7.4 is perfectly normal, so we call that normal. And then 7.41 to 7.45 is what I call basic normal, base normal. And another term for base is alkaline, right? Alkaline equals base. All right, so that's pH. 7.4 is perfectly normal. 7.35 to 7.39 is acid normal. 7.41 to 7.45 is base normal. And that's where you get that range that you probably all learned about, that 7.35 to 7.45, right? I just want to make sure we're all okay on what perfectly normal pH is, because that's a very important when it comes to talking about compensation, which we probably won't get to today, but all right. Now, moving down, the next value is the CO2. Does anyone know what the normal range for CO2 is in blood?
Is it 35 to 45?
Good. Yeah. 35 to 45 millimeters of mercury because it's a dissolved pressure. Good. And what about the bicarbonate? What is the normal range for bicarbonate in the blood?
Is it 22 to 28? No, 24 to 28.
You're in the right direction. You're in the right direction. Yeah, you're definitely right in there. I think of
We
the
already
best.
know 20 is perfect.
No, so the only
Normal.
one where you get perfectly normal is pH. The rest of them are ranges. So think of the best years of your life, 22 to 26 milliequivalents per liter, or millimoles per liter. So 22 to 26, best years of your life. And then PaO2 is the pressure of dissolved oxygen, and that is 80 to 100 millimeters of mercury. Now, the PaO2 is not used for acid-base analysis, but rather it is used for oxygenation. How well is oxygen getting into the blood? 80 to 100 is considered the normal range. Less than 80 is what we call hypoxemia. At least if you are breathing room air. And greater than 100 is hyperoxia, at least if you're breathing room air. All right. So pH, PaCO2, and bicarbonate are the three values that we look at to determine someone's acid-base status. And that makes sense because we just went through almost an hour of math to get to the three variables, if you will. All right. So some additional stuff that I want you to know. I want you to remember the following. Rome. And that stands for, that's a way of memorizing the relationship between pH, CO2, and bicarbonate. All right, and so that stands for respiratory opposite. So that is to say that the pH and the CO2, because that's really what this is looking at here, right? This is looking at the respiratory component of acid-base balance, all right? So pH and CO2 are going to move in the opposite direction. So for example, if you have a low CO2, that is going to cause a high pH, right? So that is going to be more alkalotic or basic. If you have a high CO2, that is going to lead to a low pH. And remember, a low pH is more acidotic. So pH and CO2 are reciprocal to one another. They move in opposite directions. That's respiratory opposite. M is metabolic equal. And that is referring to the bicarbonate, which is a reflection of the metabolic component of acid-base balance. So you've got the respiratory component and the metabolic component. So respiratory opposite metabolic equal. So that is to say that a low bicarbonate equals a low pH. A high bicarbonate equals a high pH. All right. So a CO2 of less than 35 tends to make you more alkalotic, and a CO2 of greater than 45 tends to make you more acidotic, whereas a bicarb of less than 22 tends to make you more acidotic, and a bicarb greater than 26 tends to make you more alkalotic. All right. So before, I think what I might do is I might just conclude here, because we've gone through a lot of stuff, and I don't want to actually throw the interpretation on you all quite yet. But let me just see how folks are doing. So let me stop that. How is everybody doing so far on that?
All clear.
All clear. No questions, because this is, I mean, this is as legit as it gets. Like no punches pulled, like very high level acid base. So I just don't want anyone to feel like if they're completely lost or not getting it. I just want to make sure that I don't lose
Can
anybody.
your CO2 and pH ever be the same?
No, because the CO2 is a gas, and pH is a negative log of hydrogen ion concentration. So they're measuring completely different things. They're related. They affect CO2 affects pH, but they're different units. CO2 is you're measuring a pressure, whereas pH you're measuring a molar concentration, or it is a reflection of molar concentration. So they're two different units that you're measuring in. So yeah, they would not be the same.
When you're measuring this ABG, the arterial blood gas, is that lab work basically?
Yes. So and that's what we're going to get in tomorrow. So essentially what's going to happen is a sample of arterial blood will be drawn, and then that'll be put into a machine that will analyze it, and it will spit out those 4 values and some other things. But you'll get a little printout, and then you'll look at that, and you'll look at the pH, the CO2, the bicarbonate, and the PaO2. The PaO2 just tells you the oxygenation. So really what you'll look at is the pH, the CO2, and the bicarbonate. And whatever value you see for those 3 things, that's what you use to determine the acid-base status of your patient.
Okay. Is there any way you could do any of these in the field? Like, the respiratory rate, the oxygen, kind of just to get an idea, or it's just...
Yes, there are. So one of the one of the most important tools that we have outside of the hospital to give us some idea of what somebody's acid-base status is going to be carbon dioxide monitoring. And is there a way that we can monitor somebody's carbon dioxide levels, at least indirectly?
And tidal CO2.
Absolutely. Yeah, the EtCO2. So the EtCO2 is not equal to the PaCO2. The PaCO2 is looking at the carbon dioxide pressure in the artery, whereas the end-tidal CO2 is looking at the CO2 pressure in the alveoli. But they're very related, right? The values will be very close, generally like within 5 millimeters of mercury of each other. right? So if somebody has an end-tidal CO2 of say 40, then there's a good bet that their PaCO2 is going to be, 43 to 45, right in there, right? So that will give you some idea of what their CO2 is. And then what you can do is, okay, what's going on with this patient? What are the signs and symptoms? What's the history? What are things going on? And that can inform you to some extent what an underlying acid base abnormality may be. If you put somebody on end-tidal CO2 monitoring and their CO2 is 80 and they're not breathing, well, it's pretty clear what's going on. Their respiratory component isn't working, and they will have what's known as a respiratory acidosis. But we'll get into that a little more tomorrow. Now, with that said, there are lots of services that are now able to run labs out in the field. There are various what we call POC or point of care technologies out there. One of the more common ones is something called the iStat, which is a trademark technology where you can draw blood and run the lab out in the field. And it has little cartridges. You draw the blood, you put the cartridge in for the labs that you want to run, and then you run the labs. And so there are EMS systems out there that are running arterial blood gases on patients out in the field, that are running cardiac enzymes, that are running complete blood counts, metabolic panels, and so on. So we're seeing a lot of this technology proliferating out in the field as well. So this is becoming more and more relevant. And it's very relevant when it comes to if somebody is intubated. So if they've been intubated and they are on a mechanical ventilator, understanding ABG analysis and being able to make corrections to the ventilator strategy is absolutely critical. It's actually like a fundamental transport skill for anybody who's transporting intubated patients. So yeah, there are lots of, interestingly enough, there are actually lots of applications for ABG analysis. Good deal. Any other questions? Okay, good. So right now, I just want to make sure that everybody understands what the major things are on the ABG and what the normal values are. If everybody's comfortable with that, then tomorrow when we actually go into the analysis and start talking about the various problems, so the respiratory problems, so you've got respiratory acidosis, respiratory alkalosis, metabolic acidosis, metabolic alkalosis, and then you have what's called compensation. There are different ways the body can compensate or not, and we will talk about all of that tomorrow. But today, I just wanted to make sure everybody understands what is normal, what we're looking at, where it came from. So any other questions before we conclude today? Okay, excellent. There'll be no quizzes. I do not have any quizzes assigned for you all today. So just go back over what we've talked about today, make sure you're comfortable with things. And then in the morning, we will take a deep dive into analyzing ABGs and talking about all of the various acid-base anomalies and problems we run into. All right. Before I let you all go, though, I did not see Stephen Lopez at all. So Stephen, did you happen to log in? Or are you on with anyone or anything? Okay. Yeah, I don't see in the list either. Okay. Well, without further preamble, that's all I have for you all. So have a great rest of your day, and we'll see you tomorrow morning.
Thank you, Chris.
Thank you all.
Thank you, Chris. Have a good day.
Welcome.
Thank you, Chris.
Thanks.
You all have a great day. Thank you.